All right, beautiful people, welcome to lecture 9. We're going to get into neuron and synapses, and this is a very important lecture. And I think I could say that for every single lecture, but what I want to show you folks is how it's structured here, because our little information or trajectory page is a little different from what we usually see. So today I am going to go over some anatomy review and also with electrical signaling. I'm going to review what I did last time and then go into a little bit more detail with action potentials. And then I'm going to start talking about in details how cells and cells communicate to each other. So these will all be separate lecturettes, but I do want you folks to remember, right, big picture, to categorize these items under how cells are also communicating with one another. All right. Okay, let's kick off this lecturette. Right? Cells of the nervous system. Now, before I get into separate cells of the nervous system, I just want to do a quick overview of the nervous system in its entirety. So what I've drawn here is this line in the sand, if you will, and dividing then the central nervous system from the peripheral nervous system, right? And so we all know that the central nervous system is going to be made up of the brain and the spinal cord, which is made out of a whole bunch of neurons and their associated structures. Now, there are sensory pathways, and sensory pathways are filled with sensory neurons that have sensory receptors. And those receptors now are going to sense what is going on in our body, right? How hot is it? How much sodium do I have? How much sugar do I have? And then send via axons that information to the CNS. Okay. Now, remember, the CNS is where we integrate, make decisions. So the neurons then, right, because we are talking about cell types, so I'm kind of integrating the overall picture of the nervous system, but reminding you that these are all cells communicating to one another, collectively making a decision on what should the body do in order to maintain homeostasis. So then the cells of the CNS, then after they integrate, aka make a decision, are going to send action potentials down their axon to communicate via these motor pathways, right, to different effectors. So I can have effectors that are voluntary, right? My muscle, skeletal muscle, is a voluntary effector. Can allow me now to pull my hand away or get out of the outside, which is 107 degrees. Or I can now turn on my involuntary system, my autonomic nervous system, which is made out of sympathetic cells, right? Parasympathetic cells, deciding if I should sleep, if I should rest, if I should kick up digestion because I've already ate. And then there's something called enteric. So there's certain cells, neurons that are living in different pockets inside of our bodies, especially in the digestive tract. They can actually make decisions on their own. Okay, so that's the overview. But remember, all of these pathways consist of cells communicating to other cells. Now let's quickly review the anatomy of neurons, is because today we're going to spend a lot of time communicating from neuron 1 to neuron 2, and then also from a neuron to some type of effector, right? And this is why I made a big stink about this lab in Unit 1, is because I really need you folks to have your anatomy secure so you don't have to think about anatomy. You can just get straight to functions, right? Now, these tree-like branch structures are the dendrites, and this portion of the neuron is the cell body, right? Now, a neuron will receive signals in the dendrites in the cell body, okay? And we'll be doing that today. Now, remember, a neuron does something called integration, and it integrates at that axon hillock, aka the trigger point. I don't care what you folks call it, just as long as you know that behind the axon hillock is a mechanism, right? There's a mechanism of integration. And that mechanism is I've got a bunch of sodium voltage gated channels that sit in this area. And there are many of them. Okay. So if the charge via the messages that come into the cell are strong enough, I can now open up all of those voltage gated channels to now propagate or send that signal down to the axon. Okay. And once the signal goes down the axon, there ain't no going back. Okay, it is forward. The signal has been sent. Elvis has left the building. Okay, now today we're going to talk about myelin, and oh my God, I love myelin so much that right now I'm having chicken skin, or whatever you folks call this in America, goosebumps. Now, myelin is a wrapper. It wraps around the axon and allows us to have very, very, very fast communication. So when you have an axon that's very, very long, especially the axons that are leaving the central nervous system and going out into the effectors of the peripheral body, yes, they got to be myelinated. Otherwise, you're going to lose all the charge, because charge likes to travel laterally. More on that later. Now, this is the presynaptic neuron because it comes before the synapse. Therefore, this is the postsynaptic neuron, right? Now, what's the synapse? Let me tell you. This is going to be now what we call the axon terminal. So if you don't understand axon terminal, words are important, right? I like to take the root words of the structures that we're learning. Axon terminal means end of axon, much like an airport terminal, right? You folks, if you're walking towards your gate and the gate ends, because then you got to hop on the airplane, it usually widens out. That's the airport terminal, right? So the same thing with the axon terminal, right? The axon will end and it will have a little bulb here, right? That's the end of the axon. Now, I got to get a message from the axon terminal and then have it cross over this gap. This is called the synaptic cleft. And then I can now talk to or communicate or send a signal that's going to bind now into this cells. This is their postsynaptic dendrites. Cells or axon terminals can also now synapse with the cell body, right? So this picture, I'm just showing you the dendrite. So together, these three structures is a synapse, right? Where I have communication and how I pass communication on from the previous or the presynaptic cell to the postsynaptic cell, okay? So in review, I get an input signal. All that means is the signal that a neuron is receiving. That signal will travel through the cell body. Those are graded potentials, okay? Because let's utilize our previous lectures information. So I integrate, and that integration now happens at the axon hillock. And if I integrate and I decide to send the signal, then I will have an output signal, and that is going to travel down the axon to then cause this presynaptic neuron to release neurotransmitters out of the axon terminal. That's what we're doing today in great detail, okay? So this is the anatomy. If you don't have the anatomy, move forward. We're going to be doing the anatomy over and over again. All right, let's talk about glial cells. Now, I don't want you folks to memorize this entire slide, right? So the point I'm trying to make here with glial cells, and I'll go into more details of glial cells as we move forward, but this slide is really to just jog your memory, right? Do you folks remember from anatomy what glial cells are? And glial cells are support cells, and they communicate with neurons. They provide structure and ATP and glucose. That's what it means by biochemical support to neurons, right? And so these support cells allow and support neurons so we can have the most optimal electric conductivity, right? So let's talk about some glial cells in more detail. And the first glial cell is astrocytes. And oh my God, I love astrocytes. They are so amazing. And astrocytes have these special feet, okay? And these special feet they touch everything, okay? And what you folks are seeing here is a capillary bed, right? So their feet wrap around a capillary bed. And what they do now in the brain is they form the blood-brain barrier. Okay, and so they can regulate what comes into the brain and what stays out. So if you are a water-soluble pesticide, the astrocyte feet are not going to let you come in. You cannot come into the brain, okay? Now, if you already have too much sodium in the brain, these feet not going to allow any more sodium to come in. So they control substances that come in and out of the brain, except for non-polar small substances, like cocaine and alcohol. Those things can just pass by lipid bilayers, and that's why we get inebriated, right? We can't control the movement of those small lipid molecules, but everything else, small and polar, we can control. Now, look at this. Look at this. Wait, sorry, I just get really excited. Astrocytes are really amazing, but look at this astrocyte, and they're named because they are star-like cells, and they look like stars. And look at their feet. Their feet are actually even touching veins. These are veins. They touch all the blood vessels in our brain. Now, the astrocytes, another thing that they can do is they can help give glucose to neurons. So when you eat sugar, the astrocytes will take that sugar up and actually store it in their bodies as glycogen. The brain has a small capacity to store glycogen, not like our liver and our skeletal muscle cells. That's the main storage for glycogen. But astrocytes can store a little bit of glucose in the form of glycogen in the event that we run out of sugar in our bodies. Okay. Now, what the astrocytes do is they're taking that glucose and turning it into lactate, which is a type of sugar, and then giving that form of sugar to the neurons. I don't want you folks to memorize lactate, but I just wanted to be correct in the lecture, but glucose is fine, okay? Now, another thing astrocytes can do is they can take up potassium from the extracellular fluid, which is important because remember, potassium is going to diffuse out of the axons during action potential, during nerve impulses. So if the astrocytes can take up potassium for the extracellular fluid, it can help maintain proper ionic environment for the neurons. That is so amazing. Here's another picture I want you folks to see. Okay, we're going to spend a lot of time here as well. Astrocytes are also touching the synapse between a pre and post synaptic neurons, and they can help control neural communication between two neurons. This is amazing. They can help turn off communication, and they can help turn on communication by taking away neurotransmitters from the synapse, but also returning it back into the synapse. Okay, so more on that later. Now, I love astrocytes, and I think I talked too fast. I think I got too excited. So let's just write a synapse of the functions of astrocytes. Okay, so I'm just gonna write astrocyte functions. Okay, so number one, they can provide an energy source. And I told you folks that they can actually store glycogen. Not a lot, but some. And then they can go ahead and break that glycogen into glucose and then give that glucose to the neurons. Neurons need a sugar source. They need it, right? Okay. And you can put lactate if you want to, but glucose is fine for our class. Now, number two is they can take up and release neurotransmitters. I'm going to use this abbreviation for neurotransmitters, but this is dyslexia, sorry, it should be EU. Neurotransmitters, right? So they take up and release neurotransmitters, which means I can now help regulate communication, whether turning that communication off or enhancing it and turning it on. I can do that in the synapse. Astrocytes can do that. That is amazing. All right, now they can also maintain ion levels. For optimal, can be optimal membrane potentials, action potentials, so on and so forth. Okay? Another interesting thing the astrocytes can do is that they can release precursors for ATP production to the neuron. Release ATP precursors for neuron ATP production. So as you folks can see, astrocytes are very important in maintaining optimal productivity of neurons. Okay. All right, let's move on. Let's talk about myelin. Oh, I love myelin too. Okay, so myelin is an insulator. Because remember, in the axon, we are transmitting electronic signaling. Okay, those signals like to move laterally. All ions like to diffuse down their concentration gradient, but we can't have them move laterally. We need them to move forward, okay, down the axon. So we have to insulate or wrap up the axon so we don't lose those ions, okay? So this is myelin. It's a protein fatty sheath, and we have different cells that make myelin. And in the brain, those are those oligodendrocytes that you learned in anatomy. And then in the periphery, aka if this is a neuron, then is traveling to my skeletal muscle, I have something called Schwann cells that are now also doing the same thing, wrapping this protein fatty sheath along the axon. Okay. Now, it's not critical that you're remembering oligodendrocyte and Schwann cell. I will be saying Schwann cell a lot, but just long as you know that there are cells that make myelin, then we're good. And just as long as you know what the function of myelin is, then we're good too. One structure that I do want you folks to pay attention to, and you will need to understand what's going on in here, are these structures, these spaces that are not wrapped by myelin. And it's labeled in our diagram here, but it's all micro mini. So I'm going to write big. Node of Ranvier. Ranvier, okay? It's French. Now, these nodes are unmyelinated spaces, and they will play an important role on how we now have action potentials travel down the axon. So I'll go over the mechanism of that in our next lecturette.